Well, welcome everyone. I am Tracy Smith, the Collaboration, Education, and Outreach Coordinator for the Center for Integrated Cellular Analysis here in New York City. In this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. In today's seminar, we will focus on a new method that performs multiplexed and multimodal single-cell perturbation screens using the RNA-targeting Cas13 complex. This method, and I love this acronym, CaRPool-seq, enables simultaneous perturbation of multiple genes and can identify genetic interactions that enhance or suppress cellular responses, including AML differentiation phenotypes. As always, you'll be able to ask questions at the end of the talk. You can either raise your hand and I'll ask you to unmute yourself, or you can type your question into the chat and I will read it to the speaker. Today's seminar will be presented by Harm Wessels, a postdoctoral researcher in the Sanjana and CICA labs at the New York Genome Center. He received his PhD in biology from Humboldt University in Berlin while working at the Berlin Institute for Medical Systems Biology and the Max Delbruck Center. There he focused on RNA-centric questions, performing transcriptome-wide identification of mRNA interacting proteins in Drosophila and generating maps of functional RNA cis-regulatory elements. In the Sanjana and CICA labs, he has led the development of a novel RNA-targeting CRISPR-Cas13 toolkit for forward genetic screens, Cas13 therapeutics, and Cas13 perturbation multi-omics single-cell profiling. And with that, Harm, I will hand it over to you.
Thank you, Tracy, for the nice introduction. Yeah, so let me just see to make this full screen, change it to my laser pointer. And yeah, so I'll just one more time. I need to minimize the videos here so I can access the full screen. Yeah, thank you for the introduction. Today I'm going to talk about our new method, which allows us to perform multiple perturbations in single cells. And this is a method that uses CRISPR-Cas13 as opposed to traditionally Cas9. And but before I really go into this, what we are currently doing in the field of single cell biology is we're trying to make a map of the human body, which is composed of trillions of cells and more than 200 different cell types. And each of the cell types is characterized by the expression of a unique set of more than 10,000 genes, which can be protein coding or non-coding. But in the end, what we ultimately want to know is we are interested in what is the function of each of these genes. And specifically, we can test for the function by, for example, perturbing a gene and then ask what are the phenotypic consequences associated with this perturbation. And we can do this in one cell type, which would basically mean that we need to do 10,000 tests. And obviously, right now, we can do this, we have the tools, but then at some point, there's going to be a follow-up question where the question is going to be, how does the function of one gene depend on the function of another gene? Which you see becomes a quadratic problem, so we need to have efficient methods to do this systematically. Full CRISPR screens have really allowed us to look at one phenotype and score how do all genes in the genome, in a genome-wide fashion, contribute to this one phenotype. So the way this usually works is we design a couple of guide RNAs that are targeting, for example, the promoter region or the coding region of a gene. We make a library out of that, generate lentivirus, and introduce this into Cas9 expressing cells. And we take a snapshot of this to get an idea of what is the input representation of all our guide RNAs. And then we apply some sort of phenotypic selection, and the easiest of which can, for example, be growth. So the cells will proliferate, and some of them will have a growth advantage depending on the perturbation, and others will die. And then at the end of this experiment, what we usually do is we compare which guide RNAs are present at the end and compare this to their input representation. And then we can, for example, see that certain genes or certain guide RNAs would be enriched while others be depleted. This is a great assay to really look at all genes for one particular phenotype. And what we basically do in this method is we order all our perturbations or all our cells along a one-dimensional axis. So here, for example, we have two perturbations that positively contribute to one phenotype, and we have another perturbation that has a negative impact. But what this also means is that we actually look only at one dimension, and very likely, in many cases, we are actually underestimating the complexity behind these changes. So very likely, points that are close in one dimension might actually be very different if we look at all dimensions at the same time. One way to overcome this is, instead of just looking at one phenotypic readout, we can profile content-rich phenotypes in single cells, like, for example, single-cell RNA-seq, which maybe allows us to really capture this complexity in our response. And then we can use dimensional reduction algorithms, which allow us to group cells with a similar response, and then we can make inference based on that. There have been a couple of methods established over the past 5-6 years, which really allow us to combine pooled screens with single-cell RNA-seq readout, which really allow us to perform content-rich phenotyping at scale. Usually how we do this is we have a cell line or cells that express a Cas9 effector protein. We introduce guide RNAs, like, for example, a single guide RNA into these cells. We select these cells, and they are perturbed. We isolate individual cells, and then we profile the gene expression of these cells along with trying to capture a guide RNA. And only if we capture this guide RNA, we actually know the label of the perturbation that we put into the cell. And in the end, what we try to achieve is we want to group individual cells based on their perturbation, and then we hope that there are gene expression patterns that are specific to each of the perturbations that we want to study. When I started my postdoc, I was really interested in looking at another type of CRISPR proteins, and specifically Cas13. And similar to other CRISPR proteins, Cas13 is part of the adaptive immune system from bacteria and archaea. And usually it's characterized by the presence of RNase domains, like these HAPN domains, which allow us to find these in metagenomic searches in bacteria and archaea. And usually Cas13 proteins do not come alone, but they are associated with other Cas proteins and also with CRISPR arrays. And once a CRISPR array is expressed, which is a multicistronic RNA, which has multiple direct repeats and multiple spacer RNAs, Cas13 can cleave this array into multiple mature CRISPR RNAs, which then can get loaded into the protein itself. And the protein can identify the target based on RNA-RNA hybridization and then degrade the target RNA. Since the discovery of Cas13, there has been like a similar explosion in what we can do with this type of protein compared to Cas9-based approaches. And the most basic one is maybe to look at RNA knockdown, but right now we are actually able to, for example, detect RNA viruses or to do site-directed changes on mature transcripts, like, for example, the removal of M6A sites or whatnot. But for the purpose of this talk, I want to focus on the ability of really performing a good RNA knockdown. So why should we care about RNA-targeting CRISPR proteins? So one thing that I think is important, if we have the ability to target RNA directly, we can actually target something that is not necessarily straightforward using our DNA targeting alternative. So for example, there are about 10% of all genes in the human genome which actually are present in a bidirectional promoter. This means there are two genes in close proximity that go off in two different directions. So here in this example, there's, for example, a protein coding gene on the left-hand side and the link RNA on the right-hand side. Using Cas9, we can perturb the protein coding gene, but very likely we are not successful in perturbing the link RNA. And if we use a dCas9 effector protein, like, for example, in CRISPR inhibition, the radius of action is likely larger than the distance between the two different genes. So we cannot actually unambiguously target this locus without affecting those genes. So in here, you can imagine that targeting the RNA directly would actually enable us to be specific for either the mRNA or the link RNA. In addition to that, there are other features that are not even present in DNA space. So you can, for example, imagine if we are able to target RNAs, we can target individual mRNA isoforms based on the unique junction between exons, or also certain types of non-coding RNAs that have backsplicing junctions that are definitely unique in only the RNA space. But how can we actually perform pooled CRISPR screens with a single cell readout? And I think the biggest bottleneck that we had to overcome to make this happen is that we had to find a way to actually capture the guide RNA, because only if we capture the guide RNA, we are able to tell which perturbation we introduced into a single cell. And traditionally, like in Cas13, the protein that we are working with, the CRISPR RNAs are composed of a direct repeat at the 5' end and a spacer RNA that is variable on the 3' end. So this means also that there's no constant region that we can use in order to perform reverse transcription, and we need to perform reverse transcription in order to capture the guide RNA in RNA space. So we thought of a couple of ways of how we can do this, entertaining ideas for direct capture, where we would directly add the reverse transcription handle 3' to the spacer RNA. And we also thought about leveraging Cas13's processivity of being able to cleave a polycistronic CRISPR array into multiple mature CRISPR RNAs, and then use one of these positions to encode a barcoded guide RNA, which can tell us the combined identity of all the perturbations we introduced. And here, what we can do is we have a lot of flexibility. We can add a barcode together with a reverse transcription handle and use this at the end of an array at the beginning, or we can, for example, add an additional PCR handle, which may be beneficial for the recovery of the barcode. The first thing we wanted to understand is, like, does any of our ways of how we either think about direct capture or indirect capture, does this affect our ability to perform efficient knockdown? What you're seeing here are the results from an experiment where we targeted CD46, and we read out the efficiency of targeting CD46 using flow cytometry with an antibody against CD46. And what you see here is that if we target CD46 with the standard guide RNA, we have robust knockdown, and the same knockdown we have if we target with a guide RNA that is enabled for direct capture or for indirect capture, with a very similar degree of knockdown. And this is not only the case for CD46, but we did this for 3 different targets, and in each case we see robust knockdown, almost independent of which way of guide RNA capture we encoded, either for direct capture using 10x's feature barcoding capture 1, or any of the indirect capture methods. But what was really important, besides showing that we can knock down a target, even more important than that is our ability to capture the feature that we are interested in. And what you see here is an amplification plot or a gel that shows us that we can detect the feature we are interested in direct capture here, but also, and most pronounced, using indirect capture, and especially if we add an additional PCR handle just upstream of the barcode, which likely tells us that reverse transcription may not be complete, but adding a PCR handle makes us independent of this success. So in the end, we decided to move forward using Cas13 RNA Perturb-seq, or CaRPool-seq, how we call it, in the way that I'm describing here now. So we're taking advantage of Cas13 ability to process a CRISPR array, and we use a single barcoded guide RNA that is surrounded by an RT handle and a PCR handle that tells us the combined identity of all perturbations. We found that there was no significant reduction in target knockdown, and we also see that this obviously enables very efficient combinatorial targeting. And importantly, all these features within this array, they are tightly linked. And since we have full flexibility about which PCR handle and which RT handle we are using, we can easily think of ways on how we can get a specific amplicon for our cDNA library, but also how to make it compatible with other single-cell modalities that we are maybe interested in. As a proof of principle, we started knocking down, to perform CaRPool-seq to target cell surface proteins either alone or in combination. And for this, we used 10x's feature barcoding, where we used the feature barcoding handle in order to capture the barcode we are interested in, and we combined this readout with CICA, which gives us an idea about how good are we in knocking down the cell surface proteins, because with this we can sensitively measure the degree of knockdown of CD46 or CD55 and CD71. And just to show you, like, our having the flexibility of choosing the PCR handle and RT handle, we can really precisely get the cDNA amplicon that we are interested in alongside with other cDNA amplicons that allow us, for example, to do cell hashing or to get cell surface protein information. What we noticed when we looked at these 29 different perturbations that we encoded, we saw that we are able to specifically recover the bar-coded guide RNAs in scenarios where we use a single guide RNA, like here using a non-targeting guide RNA, or in scenarios where we encode 3 different guide RNAs. And in each case, the recovery efficiency was nearly equal. Here, what we're looking at is we're looking at the pseudobot gene expression profile of all cells that have received a non-targeting guide RNA and cells that have received a triple copy of CD55 targeting guide RNAs. And what you can see is that overall, there's not a lot happening, but CD55 is specifically dropping out from this population. And this was not only true for CD55, but we saw the same for CD46 and CD71. That in each case, we see specific knockdown of the target we are interested in. And importantly, if we just, for example, look along this row, we see that these 3 individual perturbations, they actually map or like congruently map together if we do a triple perturbation in using these 3 different guide RNAs. And while this is maybe on the RNA level not the most pronounced, if we look on the protein level, here we have a way better idea of how strong is the knockdown, because using the ADT levels, we can sensitively say how strong or what is our ability to knock down the target. So here you see the UMI levels of the 3 cell surface proteins that we targeted. So CD46, CD55, and CD71. And what you can see in each of these violins is that once we start targeting one particular gene, like CD46 with 1, 2, or 3 guide RNAs, in each case, we have a strong depletion of the target gene, almost by an order of magnitude. And this was the same for CD55 and the other 2 genes, and not only targeting them alone, but also by targeting them in combination, like here on the very right of this plot. And we can use this information and then start clustering our single cells based on the profile of the cell surface proteins that we recover. And if we do so and then label the cells by the bar-coded guide RNA that tells us which guides we actually used in this perturbation assay, then we can see, for example, in this blue cloud here on the top right, that these are all the cells for which we could make the assignment that we have a triple knockdown of 3 individual targets. And importantly, it seems to be independent what is the actual orientation within each of these arrays, because we can swap around the order of the guide RNAs. Another way to look at this are these feature plots, where here, for example, I highlight in the top row in black, what are the cells that have any kind of array with at least 1 representation of CD46. And you see that on the entire right half, we see cells that have CD46 guide RNAs. And if we then ask, what are the cells that still show mRNA or protein expression, we can appreciate this mutually exclusive relationship between cells that have that carry the perturbation and cells that still show the target expression. And this is the same for CD46 and the other 2 target genes. So this tells us that indeed we can perform single and combinatorial perturbations in single cells using Cas13. But the question was, like, how good can we actually do this? Like, how good is CaRPool-seq compared to existing methods, like, for example, direct capture Perturb-seq? And while there's a number of differences, the key point that I really want to focus on is that if we want to introduce a triple perturbation in CaRPool-seq, the advantage we might have is that we actually need to only capture 1 single feature, which tells us the combined identity of all our perturbations. And if we wanted to do the same using direct capture Perturb-seq, we need to express 3 different guide RNAs from 3 different promoters. And we also need to capture 3 different guide RNAs if we want to know which perturbations we introduced. And this can be challenging, especially if we move from 1 perturbation to multiple perturbations. And just to illustrate this, over the years, we come to we have an experience that about 80% of the times, we are actually able to confidently assign a single cell with a correct guide RNA. So this is a probability that applies to 1 guide, but now we need to multiply this probability the more guide RNAs we need to capture. And in the scenario of needing to capture 3 guide RNAs, we are easily left only with half of the cells that we put into the experiment So suggesting that this is maybe not the most efficient way to encode our perturbations. And this is the number of cells under the assumption that all our perturbations actually are active in the cells that we assign. So that's why we thought of comparing CaRPool-seq to direct capture Perturb-seq using 3 different CRISPR nucleases. And here were Cas9 proteins. So here we compared CaRPool-seq against direct capture Perturb-seq using Cas9 nuclease. So here we are able to perform knockout of the target genes, and 2 different versions of CRISPR interference, one which uses a CREP domain and one which uses a CREP domain together with MECP2. And what I hope you can appreciate on this plot is that really all the 4 different methods, they nicely integrate and the cells are nicely mixed. So suggesting that all different CRISPR effectors, they can actually lead to very similar perturbations. So now if we split this by our different CRISPR effectors, then we see that all the different, and we label these cells by the perturbation that we can assign based on the guide RNAs that we recover, we can see that all the different perturbations that we're interested in, ranging from single to multiple perturbations, are present in all 4 cases. But what we wanted to understand then next was how comparable is the knockdown in all these cases. And in order to illustrate this, we focus on the population that for which we were confident that all 3 perturbations for all 3 cell surface proteins were present. And if we compare this, then we see that using Cas13 or any of the Cas9 based approaches, we see a very similar degree of target perturbation on the protein level, which is great because this means that it doesn't really matter which CRISPR effector we use. In each case, we get a strong and robust target knockdown. But importantly, what we also noticed is that using direct capture Perturb-seq, we found that it was increasingly getting harder to assign the correct number and the correct identity of guide RNAs, the more guide RNAs we were interested in. So what we actually did is we performed this experiment using cell hashing. So we knew exactly for which cells we expected 1, 2, or 3 guide RNAs. So we can quantify in how many times we correctly assign the correct number of guide RNAs. And we found that if we, for example, require the assignment of 3 guide RNAs, we could only use about 50% of the cells to do so. And this was very much in line with the extrapolation that I just showed you on the slide before. So the question is, okay, this apparently works, and CaRPool-seq is like really well suited in order to study higher order perturbations in single cells, but what can we use CaRPool-seq for? So we thought of using CaRPool-seq to learn more about acute myeloid leukemia, and specifically acute myeloid leukemia is characterized by having myeloid progenitor cells that are continuously proliferating. And actually, there's an AML differentiation block which really inhibits the ability of these cells to mature and then stop being proliferative. So in a normal myeloid differentiation pathway, what we usually would have is these progenitor cells would differentiate, and we could recognize this, for example, by the expression of cell surface proteins like CD11b, that these cells are differentiated and they're not mitotic anymore. So in order to treat AML, one of the therapies that have been proposed or that are also being used is to overcome this differentiation block. So to basically take these cells that are continuously proliferating and make them a mature cell type. And we can do this by, for example, targeting a target gene like KDM1A. And if we do so using Cas13, we see indeed these cells now strongly express CD11b, and which is much higher than, for example, cells that received a non-targeting guide RNA, suggesting that indeed, if we perform a genetic perturbation, we can see that these cells undergo differentiation. So we use this framework, and specifically here we used TH1 cells, which is a model for AML, and we generated a small library of about 5,000 guide RNAs targeting 440 genes, which are involved in AML differentiation. We introduced this library into TH1 cells, and after a couple of days, we started the induction of Cas13 to start targeting. And after 2 weeks, we used flow cytometry and then gated on CD11b high and low populations in order to enrich for cells that show high upregulation of CD11b. And in doing so, we identified a number of genes that show really high upregulation of CD11b. And KDM1A I showed you before, but there's a number of other genes that were also able to upregulate CD11b. And these results that we found in this pooled screen using Cas13, they were very much in line with a similar pooled screen that has been performed previously using Cas9. But our question that we had is, like, does combinatorial perturbations of these individual genes that allow differentiation of AML cells, does this improve AML differentiation? In order to answer this question, we generated a library of about 400 different CRISPR arrays, and which in total included 186 different guide pairs, and each of which in 2 replicates, covering 28 different single perturbations and 158 dual perturbations. And importantly, we have here 2 independent replicates where we target, for example, the same single gene or the same dual gene perturbation, and have basically an inbuilt control because we can compare the 2 technical replicates. And we used a very similar setup like before. We used, again, Th1 cells, transduced the cells, and then started the treatment or the expression of Cas13 by adding doxycycline. And then after 5 days, we performed CaRPool-seq. And then after 8 days, we also performed another pooled screen in order to be able to compare the CD11b enrichment from the pooled screen with the results that we got from our CaRPool-seq experiment. And just like before, we captured the barcoded guide RNA using feature barcoding, and we also kept track of the expression of cell surface protein by combining CaRPool-seq again with CICA. And as I said, at day 8, we performed another pooled screen, and again bin cells that showed high expression of CD11b and low expression of CD11b, and compared the relative abundance between these 2 different bins. So in total, we looked at 22 different cell surface proteins, and what we noticed is that really in almost all of the cases except 2, and these 2 are actually 2 negative controls, we see an upregulation of CD11b. And as I said, we have 2 technical replicates, like 2 different gene or guide pairs targeting the same single gene or the same dual gene. And if we compare these 2, then we can actually see that the CD11b upregulation in our CaRPool-seq experiment shows a very good correlation across these 2 different technical replicates. And we can compare these results from our CaRPool-seq experiment, where we can measure the CD11b upregulation on the ADT level and compare these results to results that we got from the pooled screen that we performed 3 days later. And also here, we see a very good agreement of the upregulation in CD11b when we, for example, perturb the expression of KDM1A or a negative control like ataxin 7L3. So this suggests to us that we can use CaRPool-seq and quantitatively assess how gene perturbation lead to AML differentiation. So now we have we know how a single perturbation and dual perturbations influence the upregulation of CD11b as a marker for Th1 cell differentiation. So what we can do now is we can compare, or we can ask the question, how well does the mean of 2 individual perturbations explain the actual dual perturbation that we also tested in our experiment? And what we saw is that in general, there's a linear relationship between the mean of the 2 single perturbations and the dual perturbation that we measured. But what we also noticed is that there's a certain deviation from this linear relationship that we collected here in these residuals. And we saw, for example, that if KDM1A is knocked down together with HDAC3, then we saw a specific upregulation, so an enhancement of this differentiation phenotype. And if we go back to the single perturbations, we see that KDM1A alone has a strong phenotype, and HDAC3 alone has not a strong phenotype. But unexpectedly, if we combine the perturbation, then we see an enhancement of this perturbation, of this phenotype, suggesting that we have an additive or a synergistic phenotype here. And this was not isolated to KDM1A, but we also saw a similar effect when comparing this to GFI1 and GSE1. But of course, we want to understand what is the complexity and the gene expression response behind this. And I'm not expecting you that you're able to read all the labels here. I only want to show this UMAP of about 30,000 cells to show that there's a lot of structure. And we are just beginning to really understand the structure. And importantly, what we see is that there is structure that is often driven by a single member. So here, for example, we have a mediator complex that groups many of these perturbations, or EP300, or here at the bottom, here are all the perturbations that include GSE1, GFI1, or KDM1A. And even within these clusters, we can further zoom in and see that there's more heterogeneity. But to really understand the contribution of these individual perturbations, we turn to a model that has been previously published in order to decompose how dual perturbations, to decompose dual perturbations. And here, this approach uses a linear model where we try to ask how do the two individual perturbations lead to this joint phenotype. And we can collect coefficients together with residual error to get an idea how these individual perturbations work together to bring forward this dual perturbation. And we applied this model to our data, and we could largely group our data into 4 different clusters, ranging from cluster 1, where we saw strong synergistic effects, like often seeing genes in the same cluster, where we saw strong phenotypes and high equality between the 2 individual perturbations, to another cluster like here at the bottom, where we saw often that a single gene would dominate the effect and that there was low equality between the 2 individual perturbations. And we can zoom in, for example, into cluster 1. And as I said, we see a lot of evidence where 2 individual genes contribute similarly to a dual phenotype, like, for example, for 2 genes that are in the same complex. Like here, if we look at, here we're looking at 2 genes of the mediator complex, where a similar set of genes are upregulated and downregulated in the individual perturbations and also in the dual perturbation. And we see, for example, that there are also other genes that we may not directly expect, like SMARCD1, which show a very similar profile in the top upregulated and downregulated genes, grouping with a mediator complex. And we also see that, for example, GSE1 and KDM1A also share a very similar signature, with a similar set of genes being upregulated and a small number of genes being downregulated. And this is similar between the single perturbation and the dual perturbation. And on the other end of this of the spectrum, we also see examples where one gene can dominate another gene. And one example of this is, for example, EP300 when paired with KDM1A. We see that in KDM1A and EP300 cells, we see that there's a certain set of genes that is upregulated and downregulated, which are different from the genes that are regulated if we perturb KDM1A. And if we look at the dual perturbation, then we see that the response specific to EP300 overrides entirely the response specific to KDM1A. And this is true in this bulk assessment, but this is also very consistent if we look at individual cells, like you can see here at the bottom. And just cycling back to our initial experiment, where we saw a synergistic effect between HDAC3 and KDM1A. So also here we see that HDAC3 uses or leads to an upregulation of a set of genes and a downregulation of another set of genes. And this overlaps with with the genes that are upregulated if we target KDM1A, at least for the upregulation part. And we see that if we combine this perturbation, then we see an enhancement in the upregulation, but also the downregulated genes, they are still being downregulated, suggesting that both gene expression signatures are coming through in the dual perturbation. And again, we see that this is not only true in this bulk assessment, but this is also true on the single cell level. And we were not actually the first ones that really noticed that there's an interaction between HDAC proteins and KDM1A. There's actually a study from a couple of years ago which already suggested that if we combine a drug targeting of KDM1A and together with a pan-histone deacetylase, that we can actually improve the outcome or improve the treatment of AML cells. So just to summarize, what I've shown you today is our new method for performing Cas13 RNA Perturb-seq, which is basically nothing else but an optimized strategy to deliver multiple perturbations in single cells. And this is not unique to Cas13, but it can actually also be applied to other CRISPR proteins that have the ability to process an array, like, for example, the DNA-targeting Cas12 or another RNA-targeting CRISPR enzyme like Cas7-11. And really what's important here is that we have a flexible design that really allows us to enable multimodal single-cell readouts. So here, as an example, I showed you that based on the choice of using a certain set of protein of a PCR handle and an RT handle, we can specifically detect the barcoded guide RNAs alongside with other modalities like cell surface proteins. And through benchmarking, we showed that that the amount of target gene perturbation is comparable to existing Cas9-based methods. But importantly, what we also saw is that the ability to capture multiple perturbation was much improved using CaRPool-seq, because we only need to capture a single feature that allows us to identify all perturbations that we introduce. And of course, here we looked at targeting multiple protein coding genes, which is obviously great, but this is not limited at that, because we can also think about Cas13 as a way to target other RNA species, like, for example, non-coding RNAs, like, for example, link RNAs, enhancer RNAs, or circular RNAs, or also in the future, maybe applications where we use a deactivated version of Cas13. And we used this approach in the end to identify genetic interactions in AML, and we found that there is a genetic interaction between HDAC3 and KDM1A, which shows an improved outcome in AML differentiation. And with this, I want to close and really just thank the people that were really instrumental in this study, and especially like my two mentors, Rahul and Neville, that were really there to enable this project, and also Alejandro, with whom together I do a lot of the Cas13 work. And of course, I also need to mention the Tech Innovation Lab, and especially Peter and Eleni, who have now moved on to either 10x and Immunai, and were also really helpful in this project. And with this, I want to thank all of you for listening, and I'm happy to take any questions.
Thank you so much, Harm. Thanks for sharing your work. It's a great talk. So we'll move on to our question and answer section. If anyone would like to ask a question, you can raise your hand or type your question into the chat box and I can read it. And looks like we have a first question here. So how easy is it to design suitable guide RNAs for multiplexed arrays? Do you need to do a lot of optimization or large screens?
Yeah, that's a good question. And I think the first thing we have to recognize here is that the Cas13 field is maybe like 5 years behind the Cas9 field, which means that we are maybe not there where we are for Cas9. So there's a little bit more of upfront work that we have to do at the moment to really get to the best guide RNAs. And as you already pointed out, it is important that we really use guide RNAs for which we are certain that they really lead to the perturbation we are interested in. So while before this work, I was working on a different project where we used Cas13 in order to define the rules and build a predictive model that allows us to predict what are efficient guide RNAs. But obviously, like as we did in this study, if we would do an upfront screen to score individual guide RNAs and really identify the ones that are most active, that would obviously be beneficial. So we can think about ways of, for example, where we just either do an upfront screen and then do a dedicated library for a single cell assay, or we can also think about a screen where we just put all the combinations that could potentially work together and then have a phenotypic readout that allows us to select specifically for the ones we're interested in. So for example, what we could have done is also we just do a larger CaRPool-seq screen and then use our FACS strategy to selectively enrich for the cells that show the most pronounced enrichment of CD11b.
Great. Another question that I had was about incorporating other modalities into CaRPool-seq, chromatin accessibility or DNA methylation? Are these things that you see coming?
Yeah, I think that's a great question. Just because, like, I think what I really like about this approach is that we have a lot of flexibility of what are the different RT handles and PCR handles that we use in order to surround the barcode that we are actually interested in. And we can imagine that like we were able to build a combination that allows us to get a specific cDNA amplicon in order to make this compatible with cell hashing and CICA. But we can also imagine that there's another combination of PCR handles and RT handles that allow us to, for example, incorporate other handles that are maybe needed, for example, for chromatin accessibility. I haven't started that, or I haven't done those experiments, but I'm sure those are possible.
We have a couple questions in the chat about higher order perturbations, maybe like how large, how many genes do you think, yeah.
Yeah, that's another good question, and I don't have a good answer to that. Like, I've played around with arrays up to the length of 9, and I could see a knockdown of up to 9 genes. If this is still interpretable is a different question. So I think if you have a very defined question that you want to answer, I think it's fair to say that we can easily study 3, I would say, perturbations like that. But it becomes like the more perturbations we add, the harder it becomes to really interpret the results that we see.
Okay, well, thank you again. We're at the end of our time. Thank you all for your questions, and it's been a great talk, Harm. Thank you so much. As always, this seminar has been recorded and it will be available soon on YouTube and on our website, which is multimodalintegration.org. You can follow us on Twitter or check our website for the latest announcements regarding upcoming talks and events. And with that, I'd like to thank everyone for joining, and we hope to see you soon at our next seminar. Have a lovely day.